So I am very soon. I'll share the screen with you. You just hold on, and then we'll have a discussion on that. Then we'll see that how we can have the discussion on all those things. So you just wait, and then I'm just sharing the screen with you, and you will have a more information regarding the topic. Now I think the screen is on in front of you, so you will have a better understanding that the principle of food science and nutrition. Under this topic, we have decided to discuss the carbohydrate importance and classification. And you know this course is from School of Agriculture. Myself, Dr. J.K. Sharma, I'm interesting this course. The objective of discussion of this topic is that to understand the concept and importance of carbohydrate, and we'll also discuss the types, characteristics, and specialties of carbohydrate. So these all are the things those we'll discuss in the incoming topic. So first, let us have a discussion on carbohydrate. Carbohydrate is nothing; it is the most abundant class of biomolecule. You know, it's the most abundant class of biomolecule and important macronutrient also. Perform carbohydrate physiological perform important physiological functions of the body associated with pathological conditions like diabetic, mellitus, lactose intolerance, etc., etc. So it's very important that carbohydrate is one of the premier nutrient which is in general which is of very much use, and this carbohydrate is very much used in several that physiological activity, and it's an important macronutrient also because it's the first who responsible for the energy and energy production in the body. So it means that if the carbohydrate is it, if there is any deficiency of carbohydrate, it's a very bad sign that the animal or human being they started taking energy from other source. So there are certain that the pathological conditions also, for example, the diabetics and also sometimes both the type 1 and type 2 diabetes, they are also related with the carbohydrate. If carbohydrate is not able to if there is not proper that functioning of hormonal functioning inside the body, for example, if the production of insulin and glucagon, if it is not being properly produced inside the body, then we'll have several that issues to whom we are calling the diabetic mellitus and lactose intolerance also sometimes. So that these all are the functions those who are directly related with the body physiology, and these functions are very important as far as the body is concerned. So we have to understood this thing that until and unless the body is taking carbohydrate, or metabolism of carbohydrate is very much important. And you know that when the carbohydrate that to be further broken down in energy to be produced, then it the further the after the broken down of carbohydrate, we know that there is a formation of fat inside the body, and the storage of carbohydrate in the animals, we know it's a glycogen, and in the form of the glucose is stored in the liver, and so on other part of the body. So that is very important, and the carbohydrate is one of the premier nutrient which is to be treated as a macro nutrient. It's a macronutrient. It means it's required in a huge amount. Then the performing very important physiological function in the body associated with pathological conditions, and then the most abundant class of the biomolecule. It means further if we will there is no carbohydrate that the metabolite. It means the finally we have a carbon dioxide and the energy. So that is also very important. Now, carbohydrate may be defined as or saccharides may be defined that Greek, emerges from the saccharon, meaning sugar, that organic compound composed of carbon, hydrogen, and oxygen. So carbohydrate is nothing, it is just the combination of carbon, hydrogen, and oxygen. Many carbohydrates contain nitrogen and other elements, but they are not in general available. So, but they are having certain other nitrogen and other sulfur sometimes. These are few important that element they are also to be there. But in general, the carbohydrate is actually the chemical compound which is having carbon, hydrogen, and oxygen. These three compounds they are to be there if we are calling something as a carbohydrate. Then we'll have a discussion on the chemical definition of carbohydrate. That chemical definition is that polyhydroxyl having more than one hydroxyl group. It means in the carbohydrate this minus OH group. It means more than one OH group. If it is there, then we are calling it the carbohydrate. Then the functional group, it is specific group at atoms or bonds which are part of the larger hydrocarbon chain, provide a specific chemical behavior. So sometimes it's a aldehyde, or sometimes it's a ketone. That is the meaning of that thing. Then for carbohydrate, the functional group is the carboxyl group, carbonyl group, which is maybe either aldehyde group or maybe carbonyl group or keto group. So these two, the CHO and CO, these are the two groups. The CHO group is being called as aldehyde group, and the CO group is called as a carbon keto group. Then we'll see the structure of carbohydrate. That is very important. That the classification of the carbohydrate. Carbohydrate mainly classified into four different groups: monosaccharide, disaccharide, oligosaccharide, polysaccharide. These are the monosaccharide means it is only the one that the, either the keto group or the CHO group. Disaccharide means the two atoms of the carbohydrate, they are attached with some bond, that the CO or CHO bond. And then the oligosaccharide, it means more than two. And then the polysaccharide, it means it is always more than three. So these are the four category of the carbohydrate. And monosaccharides are further classified on the basis of the aldehyde group or keto group. In aldehyde group, we know that aldo monosaccharides, aldoses, and in keto group, if the CO group is there, then we call them keto monosaccharide or ketoses. We are also calling them the aldoses and ketoses on the basis of the presence of the CHO group or CO group. Then the carbon chain, on the number of carbon in the chains, we are also calling them the trioses. It means 3 carbon, tetroses, 4 pentoses, 5 carbons, hexoses, 6 and 7 heptoses. So on the basis of the presence of the carbon in the chain, we are also calling them that trioses, tetroses, pentoses, hexoses, and heptoses. Then you will see that the carbonyl group, it means there is a glucose. It means CHO over here and CO over here. Glucose is being called as a we know that it's a aldosage, and fructose is the ketosage, because they are having the CO group, and they are transferring each other if in the metabolic activity. And that is to be there in the exercise. We will see that this is a very important thing, that is there. So these are a few important things that we have to understood, that what is the difference in between the aldoses and the ketoses. So we have discussed so many things till now, and from the structure, chemical structure, which is in front of us, that the carbonyl group, they are also interchanging in each other, because sometimes the glucose is the HCO, CHO is there, and that is the aldehyde group, and further it may be changed in the if the hydrogen removed, then it may be changed in the carboket that keto group in the form of the fructose. So this is the monosaccharide change into the aldehyde change into the ketoses. That will see, we can see easily from the chemical structure. Now we have example of monosaccharides, that there are the plenty of examples, that the trioses and the tetroses and the pentoses and the hexoses. So 3C means, it means the three atom of carbon is there, and the example is glyceraldehyde in the aldoses. It means here the three C, and they are having the three number of the aldehyde group, and then the hydroxy acetate, that is the example of the ketoses. When we are talking about the trioses, it means they are having the three atom of the carbon in their chain, and then that is why we are calling them the trioses. And the example of aldoses in the trioses is glyceraldehyde, first one, and in the ketoses dihydroxy acetone. Then we have a tetrose. It means here the presence of four carbon is there, and the example is erythrose, and then the erythro erythrulose. This is the example of the ketoses, keto tetrose. Then the pentoses, we here the presence of five carbon chain is there, and the example is very that ribose and the xylose. Then in the keto group, it's ribulose and xylulose. Then we have a six atom of carbon, to whom we are calling the hexoses, and the example is glucose, galactose, mannose, and in the case of the keto, it's fructose is an example of the hexoses. So these are the few, that how the monosaccharides, they are being classified, and they are being understood. Then we'll have the other that the state chain structure of typical monosaccharide, that the anomeric, you see that the structural, that the combination, that the if the C attached with the CHO is attached in the front, and the CH2 OH in the terminal alcohol carbon, and then we have a penultimate carbon, the OH and H is also attached. These are the two. Then the epidermic carbons, that we'll see that simultaneously they are attached. So this it's the example of the typical monosaccharide, that is glucose, and it's also the hexose, and it's also a example of the aldehyde group. Then we have the assistant of D or L, that according to the orientation of the penultimate carbon, if the hydroxyl group is on right side, the molecule is D sugar, or if it is in left side, then we'll call it the L sugar. So L sugar is the example, that here the we see that the hydroxyl group is on the other side, and then it's in the reverse condition. We'll see that the OH group is in the here, on the fourth carbon chain. You will see that the OH condition is reversible. So that is the L sugar, and that is D sugar. If it is on right side, then it is the D sugar, and if it is on the left side, then it is the L sugar. So this is also on the basis of the L sugar or D sugar, that on what position the OH is there. Either it is in the right side, when we'll call it D sugar, and if it is on the left side, then we'll call it the L sugar. So these are few that the chemical structures that we have discussed. Then D or L isomerism produce minor images, that we'll see also, that in right side and left side, that the L glucose and D glucose, that we'll see that right side we have a, and that kind of that situation, where the OH is on the right side, and when OH is on the left side, then we'll see the L glucose. So this is also one of the mirror image is also, that opposite we'll see, when we'll see in the mirror, then we'll see in a different way. Then the oligosaccharides, there are several that the 3 to 9 that the monosaccharide they are attached with the bonds whom we are calling the glycosidic bond, and the raffinose, the trisaccharide, and the stachyose, the tetrasaccharide, and the verbacose and the pentasaccharide are non-digestible carbohydrate, mainly for they can cause gas. They are also sold as a bulk sweetener. They are also the bulk sweetener. So these are very good example of the oligosaccharide. So we have discussed so many good important things that what are the glucose and sugar and sugar that we have discussed thoroughly in this particular discussion, and I hope that we'll have a very good understanding. That another picture of that the D glucose, that mirror image, it means when the just opposite we'll see in the mirror. The D-glucose here is the first one, where the OH in the right side and then the OH in the left side. It's a mirror image. It's the opposite we'll see, but it's a left and right where we are. If we'll see the mirror image, it's the opposite of your. You see that in mirror we'll have this kind of that picturization of the D-glucose and the L-glucose. They are very important. That where is the isomers OH is there. So on the basis of that, we have that sense of positioning on the fourth carbon point, of course, and then in L it's on the left side, and OS is the right side. It's the mirror image, and we'll see that the mirror image is also very much important. Then we'll have a discussion on the oligosaccharide, that the 3 to 9 monosaccharide. It means here the more than two, but more than two, and about that nine, up to nine, is to whom we are calling the oligosaccharides, and they are like that the trisaccharide, pentasaccharide, tetrasaccharide, and for example, for raffinose, it's a trisaccharide, and then the stachyose is the example of the tetrasaccharide, and the verbacose is the example of the pentasaccharide, are non-digestible carbohydrate mainly found in beans, and they can cause gas because they are not digested by the general enzymatic system which we have in our body. But only in the case of the animals, it is there, because they have microbial digestion, and they are up to certain extent, they can digest these kind of those who are non-digestible, that carbohydrate for human beings, but they are to be digested up to a certain extent. Only the lignin and the few hemicellulose, that they are not digestible in the microbial digestion, through microbial digestion. But the most of these, the tetrasaccharide and verbacose, that they are too very much digestible in the ruminants, particularly, for example, the cow, buffalo, and sheep and goat. These are the ruminants, and they have microbial digestion in their digestive system. So they can easily digest these kind of oligosaccharides. But in human beings, up to certain extent, we are not able to digest them. So sometimes if we consume these things in larger amount, then they cause the gaseous formation inside the body, and sometimes there might be the infection, and it may cause certain the indigestible problem in the human being. So these are the few important that oligosaccharide, though those who are very important, and we have to understood this thing, the importance of these oligosaccharides. And in the oligosaccharide, we know that the 3 to 9, that it means that they are more than two. Every time when we have a more than two, then So we have discussed this that the oligosaccharides they are in detail that raffinose and the stachyose and the verbose are non undigestible in the case of the human beings, but they are very much digestible in the ruminants. We know that the ruminants and the ruminant those who are having the microbial digestion and they have, because of the presence of the ruminal microflora the microorganism and the protozoa and other that yeast and fungi. Because of presence of these things, the fact of we are calling the ruminal flora in the ruminants, they are very much in position to digest these all things. So that is very important that the how these are, but they can cause gases in the human beings if we by mistake if we have consumed little bit more. Up to certain extent, it's okay, but if it is little bit more that to be consumed by the human being, then they may cause the gaseous formation inside the body, and sometimes they cause infection also. So that uneasiness and indigestion is a simple situation that to be caused by them. So we have to little bit more cautious while we are consuming these things. Then they are sold as a bulk sweetener. They are available in the market and they are being sold as a bulk sweetener. Now the isomers which differs from each other only with regard to group on that the single asymmetric carbon that on a single asymmetric carbon they are the glucose and the L glucose that we'll see that this is the state where that the when the right side OH is there, then we are calling it the OH and we are calling it D glucose, and when it is in the left side, then because this is the situation when we'll see in the mirror the situation is little bit different. We'll see that the situation in mirror is different. So we have to little bit more that in mirror you will see that the mirror image it's opposite. We'll observe that the OH is in the D glucose and then the L glucose like that. Then we have a oligosaccharide that we have discussed in detail already, and then we have aphorism isomers which are differ from each other only with regard to the OH group on a single asymmetric carbon atom. So they are like that. So we have to little bit more cautious and we have to understood that what kind of that the D glucose, and that's why we are calling them the D glucose and the here the will see that the example of D glucose and the D galactose. Here the isomers is little bit different. That we'll see that the in chain we have OH and then H and then H. Here in the galactose, so these are the single asymmetric carbon atoms. So that we calling that D glucose and then D galactose. Then most of monosaccharide derives of biological importance, that what kind of that the monosaccharide derivatives of biological importance, that the oxidocin products, sugar acids, then reduction product, the sugar alcohols, and the amino group and the sugar phosphates. So these are the derivatives of the biological importance, and they are very much useful in the entire that metabolic activity inside the body of a living organism. So we have to little bit more cautious that the oxidation product like sugar acids, we know that the sugar acids are those to whom we are calling the that the propionic acid and the acetic acid and then the isovaleric acid, valeric acid, and then the butyric acid. These all are the sugar acids. Then the sugar alcohols we have also the reduction that we know that the certain that the alcohols they are also being produced. And the amino group sugars are also there where we have an S2 group attachment that the amino group is there. And then the sugar phosphates, they all are in the Krebs cycle. We'll find them the importance of all these things. So they are the derivatives of biological importance. Monosaccharides are the derivative of biological importance because lot of that the important things they are to be there. So in that case we have to understood this thing. Now the mutar rotation is very important, is a term given to change the observed optical rotation of a substance with time. Glucose, for example, can be obtained in either alpha or beta pyronase form the two forms have different physical properties such as melting point and the optical rotation. So that melting point is different. If we have two, the alpha or beta, it means the melting point of these sugars is different. When either form is dissolved in water or another may not be. So its initial rotation changes with time. Eventually, both solutions have the same rotation. Sometimes, eventually, sometimes both are having the same rotation. So, rotation is the terms given to the change in the observed optical rotation of a substance with time. It is in time. What is actually the changing? That's why the deterioration is completed because of the rotation. Then meter rotation will see that it's a very fantastic situation that how the meter rotation that the beta glucose here the beta D glucopyronase now here the alpha D glucopyronase. Initially that we'll see that the alpha that the 18.5 degrees centigrade and then initially that the temperature is about 12. So I like that we will see that the initial meter rotation because of the time and after being observed dissolving water, alpha and beta form slowly interconvert via the open chain form. So there's some an equilibrium state is reached at the contained 64% beta and 36% alpha. So that is these all are the things those who are very important and that's why here we will once again go through that the monosaccharide are the derivative of the biological importance. Oxidation products like sugar acids, reduction of products like sugar alcohols, amino sugar, and the sugar phosphates, they all are there. They all are we found in the Krebs cycle and the various that the oxidation process being carried out in, even in the formation of energy and subsequently the formation of enzymes and the protein in the body as per the need and the requirement. And then here once again the epimerism that is also very important that how we can have this. So that is why the ephemerism is very important, and we have to understood this thing that how we can have that the isomers which differ from each other only with regard to the OH group on a single asymmetric carbon atom. So that is why we are discussing these important things. And we have once again that monosaccharides are derivative of biological importance that we have discussed already. That the oxidation group, the sugar, that we know that the sugars, so acid, for example, the acetic acid, butonic acid, and then the butyric acid, and then so on isovaleric acid, valeric acid. These are the important, the short chain fatty acids. They are very much important. We know that the propionic acid is very much required for the conversion of the glucose, because the propionic is converted into the glucose ultimately in the presence of the in the presence of the certain enzymes and hormones. So that is why, and as per the need of then in every conversion we have energy production. So that is why importance is very much, and the alcohols, they are also very much important for the construction of fat and lipids inside the body. And then we know that the CHO, that is also very much required. And then we'll have all the things like that. So we know that the sugar phosphate and amino acids, we know they are the amino sugars. You know that they are very important when the protein synthesis is required, and the protein is very much, you know that for transmission of the genetic material from the next generation, that the amino sugars are very much important, and they are the integral part of in the nucleus formation, inside the cell formation, and inside the reproductive cell. So that is very much important, and we have to understood this thing, and that is why we are discussing in the food science and nutrition. Then we have a meta rotation, that is very important. That the term meta rotation is given in the change of the observed optical rotation in subsequent within time substance in the within the time. It means the same substance may be changed in different, because of the deterioration, or because of that the temperature and other that environmental effect on the substance. So glucose, for example, can be obtained in either its alpha or beta pyronase form. Two forms have different physical properties. It means they have such as the physical power melting point, they might be different, and the optical rotation also may be different. When either form is dissolved in water, its initially rotation changes with time. So it's not possible that sometimes the one form is dissolved in the water, but sometimes the other forms may not be. Eventually, both solutions have the same rotation. Sometimes the some rotation, but the temperature and boiling point are different, melting point are different. So that is why we are discussing this thing, and then we'll see that the meta rotation over here, that we will see the through the chemical structure, will further have a clarification that here the beta D glucopyronase in the first one, and then we will see that the alpha D glucopyronase. So initially here the 18.7, and here in the second one we have 112.2. So that is the difference sometimes, but they both are having the same. You'll see that the position of both are same, but only that the chain will see that the change is here only, that the OH is here, and here the OH and H. So that is the only change, that the H is over here instead of that, and otherwise by and large everything is same. Here only the difference is that here is the difference, that OH is over here and H is over here. So that is why we are calling it alpha and beta. Then we understood this thing in a different meaning. So anyway, you have to now better clarity, and we have discussed all the things. Once again, I'll go through the objective, that we have understood the concept and the importance of the carbohydrate, type and characteristics and specialistics of the carbohydrate. We have discussed thoroughly, and I hope that through the discussion you will have a better understanding, and you will have not only the understanding, you will have more clarity about the content of these monosaccharide, disaccharide, trisaccharide, tetrasaccharide, pentasaccharide, and we have hexasaccharide, and then we have a then the heptasaccharide also. So we have discussed so many things, and we know that the penta and the hexa and tetra, these are the saccharide. For example, the acetyls and the verbicose, and these so on. They are very important. They are generally found in the beans, and because of the presence of these, in human beings, sometimes they created they generate the gaseous formation in the body, and sometimes they create the position of that the tympanitis, and sometimes the bloat, and sometimes the indigestion and all. sometimes the infection also. So then we have to very much cautious while we are consuming these things because of the presence of these, because they are undigestible through the enzymatic system which we are having in the in the human being. So but in the ruminants we know that they are having the specific that the digestive system, because of the presence of the ruminal microflora inside their digestive system. So they are quite comfortable, and they can easily digest this thing in their system. So thank you very much for participating in the. So now we have completed the discussion, and I hope we'll have a better understanding in course of the thing. So thank you very much. Thank you. Thank you.